Good morning. It's April 7th, and this is your daily brief in science. Here's everything you need to know. A UK-led study points to immune cell clusters and their interactions with endothelial and collagen-producing cells as potential drivers of fibrosis in Crohn's disease, shedding light on why scarring persists in the condition. The researchers combined traditional pathology with single-cell transcriptomics and confirmed results through two independent approaches, strengthening the case for the identified therapeutic targets. Experts and patient advocates welcomed the findings as offering hope for treatments that address not only inflammation but also the lasting tissue damage caused by fibrosis in Crohn's disease. The study used archived tissue for histology alongside fresh tissue for single-cell analysis, providing two separate validation methods. A Crohn's patient, Maureen Dalgleish, who donated tissue after multiple surgeries for fibrosis, shared a personal perspective and highlights the real-life impact and potential for future therapies. The work aims to map how immune cells interact with the tissue environment to drive scarring, which could influence future treatment strategies. By validating with independent methods, the study reduces the likelihood that findings are due to a single technique or sample. The approach mirrors a broader effort to link cellular behavior to long-term tissue changes in inflammatory bowel disease. While the findings are early, they identify targets that could be explored in new therapies aimed at preventing or reversing fibrosis. Overall, the study emphasizes fibrosis as a distinct therapeutic target alongside inflammation in Crohn's disease. RSV activity remains elevated for this time of year, prompting health officials to extend the immunization window later into spring. The American Academy of Pediatrics backs monoclonal antibody immunization for infants, with a CDC study finding lower RSV-related hospitalizations during 2024-25 when these immunizations were available. The extension aims to sustain protection as RSV remains the leading cause of infant hospitalization, even as the season shifts and overall severity mirrors prior years. While the reasons for the shifted RSV season aren't fully known, possible factors include environmental, biological, and behavioral influences such as timing of doctor visits and indoor climate. RSV is highly contagious and spreads through droplets and contact, with symptoms often resembling a cold but capable of severe illness in infants and people with risk factors. The virus spreads via droplets or direct contact. While symptoms can mimic a cold, RSV can lead to serious disease in vulnerable populations. Public health leaders emphasize responding to real-time disease trends rather than sticking to a fixed calendar, highlighting the need for ongoing surveillance and access to RSV monoclonal antibodies while RSV circulates. Despite the seasonal shift, the CDC notes that RSV's overall severity remains comparable to previous years. Monoclonal antibodies are a relatively new tool and are considered game changers by pediatric health leaders, though some regulators are reviewing RSV shots. Each year, about 2 to 3% of infants under 3 months are hospitalized with RSV, illustrating the ongoing impact of the virus. While the exact cause of RSV's persistence isn't clear, extending immunization access is a key response to continued transmission. Massachusetts data show Hampton County has the highest RSV-related emergency department visits, followed by the Berkshires, Hampshire, and Franklin counties. A Canadian research team from McMaster University and Sick Kids has found that oligodendrocytes, cells usually supportive in the brain, can switch to promoting glioblastoma growth by signaling to cancer cells. The central finding points to the CCR5 receptor as a key part of this communication, opening the possibility of repurposing the HIV drug Maraviroc to interrupt tumor-supporting signals. The study, published in Neuron, is led by Kui Jai and Nick Mikolajewicz, with co-senior authors Sheila Singh and Jason Moffat guiding the work. This builds on prior work showing cancer cells co-opt developmental brain pathways to spread, reinforcing a shift toward disrupting tumor communication networks. Support for the work comes from the 2020 William Donald Nash Brain Tumor Research Fellowship and the Canadian Institutes for Health Research, with Singh and Moffat holding prominent chairs. The researchers frame glioblastoma as an ecosystem, suggesting rapid clinical translation is possible through repurposing an approved HIV drug, In laboratory models, blocking signaling from oligodendrocytes to glioblastoma cells significantly slowed tumor growth, indicating the tumor's ecosystem can be manipulated therapeutically. The findings add to a growing view that targeting intercellular communication could complement direct tumor-killing strategies. The work lays groundwork for future studies to test CCR5 blockade in more clinical settings and to explore how disruption of these brain cell signals affects tumor progression. A single workout can alter brain activity in real time, and researchers have now measured it directly. In a study led by Voss, a brief cycling session after learning influenced fast neural signals tied to memory, observed through implanted electrodes in epilepsy patients. This is the first clear demonstration that exercise can modulate brain electrical activity in a way that affects memory processes. The work uses high temporal resolution recordings, addressing limitations of standard brain scans and providing direct electrophysiological evidence. After the cycling bout, researchers saw changes in ripples, fast neural events linked to memory consolidation. Beyond memory, a single workout boosted focus for up to 2 hours and increased dopamine levels, contributing to broader cognitive and mood effects. The study involved 14 participants across a multi-university collaboration, including Iowa, Wisconsin, Ohio State, Austria, and Neuromatch Academy, and relied on post-exercise measurements of SWR and related activity. Findings help explain how timing and intensity of exercise influence memory consolidation, especially when exercise occurs shortly after learning. This provides a biological mechanism for exercise's memory benefits and supports public health messaging on aging. Unlike prior work that primarily tracked hippocampal blood flow, this research shows direct electrophysiological changes in brain activity due to exercise. The results suggest that even short bursts of activity can benefit memory processes, and that exercise may strengthen memory-related brain regions over time. A new grant is fueling CRISPR in a box, a hands-on teaching kit that lets students edit genes and watch DNA change in real time, with plans to expand to more Delaware schools and beyond. The funding supports growth of the toolkit, enabling more students to directly observe gene edits and work alongside scientists. Delaware's congressional delegation says the investment helps prepare the next generation of innovators. A central goal is to reach students without advanced STEM resources by partnering with local schools, colleges, and community organizations across the state. Plans call for broader partnerships with schools and national partners to reach students from diverse backgrounds and create paths to college and STEM careers. Teachers report increased student confidence, curiosity, and excitement about science, with leaders noting the program helps students see themselves in science. Officials frame the expansion as strengthening Delaware's leadership in science and innovation and investing in students' future opportunities. The program is aligned with a national context that recognizes CRISPR in a box as a trusted teaching tool, supported by NSF funding and national honors, highlighting Delaware's role in innovation. CRISPR in a box has drawn national attention and NSF support and is viewed as a leading STEM innovation in the United States. The grant funds expanded teacher training, more classroom support, and new curriculum modules on data analysis, ethics, and medical applications, with partnerships with Delaware colleges to sustain engagement. The expansion will add more teacher training, classroom support, and modules on data analysis, ethics, and medical applications of gene editing. The Learning Lab aims to make advanced science accessible, boost student confidence, and inspire STEM careers, with educators noting the program has already reached hundreds of students. This has been your daily brief in science. To read more about these stories, follow the links in the episode bio. You can also subscribe to these updates via email at www.brief.news. For more daily podcasts about the topics you love, visit brief.news/podcasts. Tune in tomorrow. We'll be back with everything you need to know.